Hello. In today's video, we shall learn in detail about green alga Chara. By the end of this video, you will have a clear understanding about its characteristic features, classification, structure, and both vegetative and sexual mode of reproduction. Let us begin with the characteristic features of Chara. Chara is a multicellular freshwater green alga. These algae are often encrusted with calcium carbonate, making them strong and are therefore known as stonewort. Thallus of Chara is macroscopic and is easily visible with the naked eye. The alga is multicellular and profusely branched. The plant body is a gametophyte. Thallus of Chara is differentiated into a well-developed root-like structure called rhizoid and the main axis. Chara anchor themselves to the substrate with the help of rhizoid. The plant body has a stem-like and root-like structure, but true leaves, flowers, and seeds are absent. The main axis is cylindrical in shape with nodes and internodes. From the nodes, small leaf-like structures are arranged in whorls around the main axis and are known as branchlets. The thallus shows the presence of dimorphic branches. The long branches are of unlimited growth and the short branches are of limited growth. Chara shows both vegetative and sexual modes of reproduction. By the way, if you're getting some value out of this video, please like and share this video so that all of us can learn, unlearn, and relearn together. Also, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Next, let us learn about the classification of Chara. Chara belongs to kingdom Plantae, division Charophyta, class Charophyceae, order Charales, family Characeae, and genus Chara. Next, let us understand the structure of the plant body of Chara. The thallus of Chara can be differentiated into rhizoids and the main axis. The rhizoids are white, thin, elongated, thread-like structures. They are multicellular, uniseriate, and are characterized by the presence of oblique septa. These septa originate either from the base of the plant or from the peripheral cells of lower nodes present on the main axis. Rhizoids play an active role in absorption of minerals and water. Apical growth is seen in rhizoids. They reproduce through vegetative propagation with the help of bulbil or by the formation of secondary protonema. The main axis is tall, erect, and branched. The branching system in Chara is very complex. The main axis shows the presence of nodes and internodes. Nodes are portions along the main axis where branching occurs. Internodes are regions between the nodes. The internode consists of the axial cell or internodal cell and the cortical cells. The axial cell is elongated, centrally located, cylindrical cell. The internodal cells have a large central vacuole, many nuclei, and numerous discoidal chloroplasts. In corticated species, the internodal cell of main axis is surrounded by vertically elongated, narrow cortical cells which originate from the node. In E-corticate species, the cortical cells are absent. In Chara, the node is made up of two cells and is surrounded by a few peripheral cells. Three types of structures arise from the node. They are branches of unlimited growth or axillary branches or long laterals, branches of limited growth or primary laterals or branchlets, and stipulodes. The long laterals have long branches with nodes and internodes. They are branches with unlimited growth and arise from the older nodes. They have whorls of primary laterals at the nodes. The primary laterals are branches with limited growth emerging in circular pattern from the nodes of branches of unlimited growth. They are also known as branchlets. Each branch forms a number of nodes and internodes. Secondary laterals, that are hair-like structures, arise from the nodes. Sex organs develop from the lower nodes. Stipulodes are unicellular outgrowths arising from the lower nodes of branches of limited growth. When the number of stipulodes are equal to the number of branchlets, the stipulodes are known as unistipulate. If number of stipulates is double the number of branchlets, then the stipulodes are known as bistipulate. When stipulodes are arranged in a single row, they are termed as haplostephanous. When arranged in two rows, the stipulodes are known as diplostephanous. Next, we shall study about reproduction in Chara. Chara reproduce both by vegetative and sexual methods. Vegetative reproduction may take place with the help of bulbils, amorphous bulbils, amylum stars, and secondary protonema. Bulbils are small, spherical, or oval bodies. They develop on the rhizoids or on the shoot nodes. When they detach from the parent body, they germinate into new thallus. Amorphous bulbils are cluster of irregularly shaped cells formed on the lower nodes of the main axis or on the rhizoids. In adverse conditions, when the parent plant dies, these bulbils act as peronating bodies. During favorable conditions, they regenerate into a new thallus. Amylum stars are multicellular aggregates which are star-shaped. They are found on the lower node of the main axis and are filled with amylum starch. Secondary protonema are thread-like or filamentous structures formed in the basal cells of the rhizoids. They too give rise to new thalli or new plants. Next is sexual reproduction. Mode of reproduction in Chara is of advanced oogamous type. Male sex organ is known as globule. It is spherical in shape and color varies from yellow to red. Female sex organ is known as nucule or oogonium. It is oval in shape and greenish in color. It is present on the nodes of the branch of limited growth intermingled with secondary laterals. Nucule is always found singly above the globule. Species of Chara are either homothallic, that is monoecious, or heterothallic, that is dioecious. Each mature globule shows the presence of 8 outer curved plates called the shield cells. From the inner side of the shield cells arise a central rod-shaped structure known as the manubrium. The distal end of manubrium forms a globule-shaped structure called the primary capitula. The primary capitula develops into the secondary capitula. 2 or more antheridial filaments arise from the secondary capitula. It has 25 to 250 cells, which are known as antheridia. Each antheridium forms biflagellate, coiled, uninucleate anthrozoid. The female sex organ, Nucule, is oval in shape and is borne on a small stalk. It consists of a central cell and a large egg at the top. The whole structure, except the tip, is covered by spirally twisted tube cells, which are 5 in number. At the tip of the nucule, a group of 5 cells called corona cells are present. At the time of fertilization, the spirally twisted tube cells just below the corona separate to form 5 narrow slits or openings. One of the anthrozoids withdraws its flagella and makes its entry into the nucule. Anthrozoid comes near the receptive spot of the egg and fuses with it to form the zygote. The zygote later develops into a thick-walled oospore which gets surrounded by 4 layered cells. At the time of germination, the nucleus of oospore migrates towards the upper region. It undergoes meiotic division, resulting in the formation of 4 haploid nuclei. A septum divides the oospore into 2 unequal cells. The smaller upper cell or distal cell is the lenticular cell with a single functional nucleus. The lower large basal cell has 3 nuclei which later degenerate. The oospore wall ruptures and the lenticular cell comes out and divides mitotically by a vertical wall to form a large protonemal initial and a small rhizoidal initial. The protonemal initial develops into a primary protonema which differentiates into nodes and internodes. The rhizoidal initial gives rise to the rhizoids. Secondary rhizoids develop from the lower node of the protonemal filament. These structures form the new plant body. Finally, let us summarize the life cycle of Chara. The filamentous green alga Chara reproduces both vegetatively and sexually. Vegetative reproduction takes place by different structures such as bulbils, amorphous bulbils, amylum stars, and secondary protonema. Chara shows advanced oogamous type of sexual reproduction. The male sex organ globule gives rise to the antheridial filament from which arises the anthrozoids. The female sex organ, Nucule, forms the oogonium which contains the egg or ovum. Fertilization of the anthrozoids and ovum results in a diploid zygote which develops into a thick-walled oospore. During favorable condition, the oospore undergoes meiosis and germinates into the primary protonema, which grows into a plant body. So today we have discussed about Filamentous alga Chara in detail. I have some practice questions for you. Please share your answers in the comment section below. You can pause the video and write down the questions if you like. Thank you so much for your time and participation. If you haven't subscribed to my channel yet, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Also, if you want to discuss any particular topic in biology, please mention that in the comments section below. I'll see you there. Goodbye. All the best.